Presented by Caltech.
Greetings and welcome to this evening's Watson Lecture, to be delivered by my colleague, friend, and recently collaborator in the Division of Chemistry and Chemical Engineering here at Caltech, Professor Tom Miller. So as is customary, I'd like to give you a few words of background about Tom. He was born here in California in San Diego, but predominantly hails from the Lone Star State of Texas, having grown up there and also attending college in College Station at Texas A&M University. So he grew up an Aggie, and it was there that his interests in using computational approaches to explore problems in chemistry was first kindled, and that was under his undergraduate Professor Mike Hall at Texas A&M. Tom was a very successful undergrad, and under a very prestigious, with the support of a very prestigious Marshall Scholarship, was able to head off to the UK for graduate school. He initially earned an MPhil at University College London and then a DPhil from Balliol College at the University of Oxford, where he worked under David Clary, again, working in the area of theoretical approaches in chemistry. He did a short postdoc there in the UK, but shortly thereafter returned to the US and moved to UC Berkeley as a postdoctoral fellow under the tutelage of Bill Miller and David Chandler. And he was such a spectacular individual that we were delighted here at Caltech in 2008 to bring him back as an assistant professor here in our Division of Chemistry and Chemical Engineering. Our division has a spectacular history in theoretical chemistry and a spectacular number of seasoned faculty members, and Tom currently anchors the younger end of that very prestigious group and has done spectacularly well here. So he was promoted in a celebratory manner to professor in 2013, and not surprisingly, he has been awarded with a lot of accolades. So I don't want to go through all of them, but I'll just name a few. They include the ACS Early Career Award in Theoretical Chemistry. He's gotten a Sloan Foundation Fellowship. He's been a Dreyfus Teacher Scholar Award and a local award. He's also received the Associated Students of Caltech Teaching Award. Tom's a terrific teacher. As a slight personal story, I have been told by an extremely discriminating graduate student who is now a professor and was an undergraduate at Harvard College that Tom was the best teacher he ever had in any class ever. And this student would not have said that about most of our colleagues. So I hope to have put a little bit of pressure on Tom to deliver one hell of a lecture tonight. So Tom is he's a chemist, but he's a theoretical chemist. I'm an experimentalist, and there's a wonderful interplay in our division between theory and chemistry, theory and experiments that ultimately brings you hopefully closer and closer to shadows of the truth and true insight into profound problems in chemistry. The types of problems that Tom's group works in include enzyme catalysis. He also is interested in solar energy capture, conversion and storage, and also the dynamics, for example, of soft matter like biomolecules, which leads a little bit into the topic of tonight. I won't give you the full title, but my understanding is that he's going to delight us in a lecture that is an important problem in chemistry, biological, and health science. And it's a simple question: how in the heck do proteins get to where they need to go in biology, in cells, and maybe outside of cells? And that has all kinds of consequences. So I don't want to bite into any more of Tom's time. I want to deliver the lectern to him. So please, I welcome all of you once again, and please join me in welcoming Professor Miller. Thanks.
Thank you, Jonas, for that very kind introduction. Thank you all for coming, and it's a great pleasure to participate in this lecture series, to participate in the Watson Lecture Series, and to have a chance to talk to you guys about some of the research that we're excited about in my group. Now, when I tell people that I'm a theoretical chemist, they ask me what I do, and I say I'm a theoretical chemist. That's often the end of the conversation. And I think in many cases it's not always the case, but I think in many cases because there's not a very clear sense of what theoretical chemistry is. So I thought that would be a good place to start in terms of this evening's lecture. And theoretical chemists are, first and foremost, chemists. We're interested in chemistry. We're interested in the way atoms and molecules react, move, change their behavior, interact with light, interact with different sources of heat, and give rise to the different properties of materials. These are the things that we want to understand better. But instead of doing that from a laboratory perspective, we do this from a theoretical perspective, which means that we employ the fundamental underlying equations of physics, and we direct those at chemical problems. So for quantum mechanical descriptions, we use Schrodinger's equations. From classical mechanical descriptions, we use Newton's equation. And we use these equations and we solve them in the context of chemical problems. But that's not all that we have to do. One has to take those equations, and then one has to implement them actually in the context of large-scale computer simulations. So a huge amount of the work that goes on in a theoretical chemistry laboratory is to take these mathematical models and to implement them with algorithms and with programming software to describe chemical problems on large, high-performance computers. And only then, once we've developed the mathematical models and we've implemented them in adequate software, can we then use them to do the interesting chemistry, to direct them at important chemical problems, to understand fundamentally what is occurring at the molecular level, to make experimental predictions, and to help design new properties and new materials. So this is the essential workflow of what we do. We do the mathematical modeling, computer programming, and then the application of the tools. And this might seem cut and dry or relatively straightforward, but it enables us to actually tackle a huge array of really interesting chemical problems. As Jonas indicated, one of the directions in which we're very interested in is to use these theoretical methods to investigate the fundamental reactions that lie at the heart of solar energy conversion, or that lie at the heart of many biological processes associated with enzymes. These sort of reactions are hard because they're at the ragged edge of what is classical and what is quantum mechanical. They involve electrons and protons zipping around in intrinsically quantum mechanical ways, but that's happening in a complicated medium of many atoms and molecules that is better described using classical mechanics. This creates significant challenges that one has to deal with at the mathematical, the computational, and then at the applications level. Additionally, we're interested in the way that proteins fold, get targeted, behave, and exhibit their properties. And this is a similar area that involves the challenge of incorporating classical mechanics in large-scale systems and dealing with the long timescales that emerge. And a huge amount of work that goes on in my group is additionally focused on battery applications. Here you see a schematic of a battery with the anode, the cathode, and the electrode. And we're interested in investigating reactions that occur at those battery interfaces, such as the formation of unwanted dendritic morphologies that give rise to loss of efficiency, sometimes catastrophic failure of different battery materials. In the other case, we're interested in the design of better electrolytes, focusing on polymeric materials that will lead to smaller form factors, better charge density, and greater stability for a broader range of different applications. So I hope you see that from this sort of humble approach of starting with the underlying fundamental equations, developing clever algorithms, and then applying them, we can begin to work on a huge array of fantastic problems that span many areas of the interesting molecular sciences. The other point that I would make is that Caltech is, in fact, a fantastic place to do exactly this kind of research. You often hear of Caltech as a scientific playground, and I think that's a very apt description. And there's three different ways in which I think this research lives very naturally here at Caltech. You can see immediately that the work is intrinsically multidisciplinary. It lives in the Department of Chemistry, But we borrow heavily from the fields of physics, biology, engineering, computer science, mathematics, and on and on. So the ability to work at a place like Caltech that has low boundaries for these interactions and allows students, graduate students and undergraduates, to flow between laboratories in a barrierless way makes this sort of work much more natural to do here. The other obvious thing is that at a place like Caltech, you have opportunity for fantastic collaborations. And in each of these different areas, we do these theoretical predictions, we do these theoretical studies, but then we interface with experimental laboratories, like Jonas Peters' laboratory, to realize those predictions and to see what they do when borne out in the laboratory. And then finally, working at Caltech is an exciting thing for me as a theoretical chemist, because so much of what defines our field rests on the shoulders of people that have formerly, at some point, called Caltech their home, including, of course, Linus Pauling and Feynman, who underpin much of the physics and chemical principles that we employ in our research, all the way down to current luminaries in the field, including Bill Goddard and Nobel Prize winner Rudy Marcus. So I hope you see that theoretical chemistry provides an avenue to investigate a huge array of tremendously interesting problems, and Caltech's a wonderful place to be investigating this sort of research. But the point of this talk was conveyed in the title, how proteins get to where they're supposed to go. So let's turn our attention to that. Now, to understand this question, we have to think about what a cell is, and we have to think about what a protein is. And here I've shown typical examples of both. The cell, you'll see, sort of thing from any high school textbook. You see this is a eukaryotic cell. It has a nucleus. It has different compartments that are surrounded by cell membranes, basically thin layers of oil that keep the stuff on the inside in the inside. And on the right, you see a typical protein. Proteins are, of course, much smaller, so it's inflated by about 1 millionfold in comparison to the cell. And you see that protein illustrated. It's actin, but it doesn't matter. I'm just taking it as an example. You see it illustrated in terms of its individual atomic components, as well as this alternative representation here, which is the same structure, but just shown in a way that lets you see the three-dimensional geometry a little bit more clearly. Now, for the purposes of this talk, the things that you need to know about proteins are sort of listed here. They're, of course, large molecules ranging from thousands to hundreds of thousands of atoms. But there are a very special class of molecules that are formed from a sequence, a linear strand of 20 building blocks, the amino acids. And this particular representation here is color-coded with respect to the different amino acids. And as you go along the backbone of the protein, you can see the different colors indicating the different building blocks from which all proteins are made. Proteins are also extraordinarily diverse, with the human body containing approximately 20,000 different sorts of proteins that are doing all manner of different things. Now, somewhat more specifically, many proteins are made within a cell at the endoplasmic reticulum. So I've illustrated that here with these red studs, which we'll return to later, that line the exterior of the endoplasmic reticulum. And finally, the important thing to understand about cells is that even though so many of them are made at the endoplasmic reticulum, you have specialized proteins that are localized and performing their jobs in specific compartments and specific regions of space throughout the cell. So given just these simple facts, you see that there's something very interesting and challenging at play. Cells face an enormous challenge of getting proteins from where they're made to where they do their molecular jobs. And performing that function is something that they have to do to have a viable and successful cell. Now, understanding the protein delivery problem at the level of the cell seems sufficiently complicated already, but it doesn't begin to convey the actual complexity of this problem. In a sense, looking at the protein delivery problem from the perspective of this high-level cell is a bit like looking at the people delivery problem in a major city by just looking at the associated subway map. if you burrow down to the fine boots-on-the-ground level, you can see that things are actually quite a bit more complicated in both cases. So on the right-hand side, you see, of course, a busy subway station. You see this extraordinary packing density of people who are jostling and pushing and squeezing past each other to get where they need to go to perform their functions. On the left-hand side, you see a surprisingly similar picture. This is a remarkable illustration from these authors where they took the typical numbers of proteins within a cell. They knew the size of those proteins. They knew the volume of the cell. So they then used a simulation to illustrate the actual density of proteins in a typical cellular environment. And this blows the mind. It's so crowded. It's so packed. How is it getting what it needs to do, where it needs to go in this context? So there's two takeaway messages that I'd like to emphasize at this point. The first is that true understanding of the protein trafficking in cell demands a molecular perspective. You have to get down to this fine-grained level to really understand what's going on. And the second point that I want to emphasize is that the successful trafficking of proteins leads to healthy cell function. Getting things to where they need to go is paramount for this successful cell, while failure of the cellular protein trafficking, much like failure of the LA traffic system, can lead to substantial problems. And the list of problems at the cellular level give rise to these terrible diseases, which are as remarkable for their diversity as they are for their severity in many cases. So given that it's so important to understand this problem at the molecular level, let's burrow down a little bit more and see what is at play. So we return to this picture, this endoplasmic reticulum where the proteins are made, studded with ribosomes. And actually, this is a cartoon, but it's not a bad cartoon, because if you actually look at real data, it looks very similar. This is an electron micrograph on the micrometer length scale. And what you can see, again, are the folds of the endoplasmic reticulum, and you see those black dots. Those are these different studs at which the proteins are being made. And what's going on at each of those black dots is, again, a wonderful and remarkable thing. Each one of those black dots is a ribosome. And each one of those ribosomes is making protein. And each one of those ribosomes is docked, organized, onto the cell membrane, which you see we're living in the plane of there. And it docks at the cell membrane on a channel into which it feeds that newly synthesized protein. So this thing is churning out new protein, mushing it into this channel, and the channel is spanning the membrane. It's a remarkable thing. And this channel is called the translocon. It provides a transfer pathway from one side of the cell to the other side of the cell. And as you might imagine, protein gets mushed in there. There's kind of two things that can happen. Well, it can either get mushed across, in which case you get what is called a secretory protein because it's been secreted across the membrane. Alternatively, you get something that exits out into the membrane interior, forming a so-called membrane protein, again, for obvious reasons. Now, the focus of this talk is to understand membrane proteins. We see that this is the first step of the cellular trafficking problem. This is where the protein is made, and it makes this first bifurcation with respect to where it's going to be going. Now, these membrane proteins are incredibly important for all sorts of practical reasons. They comprise 30% of all proteins. Because they live in the membrane, and indeed the translocon is itself an example of a membrane protein. Because they live in the membrane, they control the flow of nutrients, energy, information, matter across that cell membrane. This huge set of 30% of all proteins that are membrane proteins are therefore incredibly important for all sorts of things associated with the flow of these different items across the cell membrane. And as a result, because of that important gatekeeper role that they perform, membrane proteins are disproportionately drug targets. Because if you want to hinder the flow of ions or matter across a particular membrane, targeting the membrane protein to turn that on or shut that off is a major target for the development of drugs. So they're important. They're prevalent, and they're incredibly important. But the bad news is they're extremely hard to study. A remarkable fact is that even though there are so many membrane proteins, and even though they are disproportionately represented in terms of drug targets, they are grossly underrepresented in terms of what we know about proteins. For example, only 3% of all experimentally solved protein structures are membrane proteins. They're very hard to work with. And indeed, there's another major bottleneck associated with the study of membrane proteins that will play a feature in this talk. It's called the expression problem. One of the reasons we have so few examples of well-studied membrane proteins is because it's hard to even get enough of those membrane proteins to perform the biochemical studies. It's hard to even make enough of these proteins. Only 5 to 10% of all membrane proteins can even be made in sufficient abundance to where we can study them using many of the most powerful biochemical techniques. And this creates a huge bottleneck that leads to this lack of understanding for this incredibly important class of proteins. So this seems bleak. These are incredibly important proteins. They're very hard to experimentally. But in a sense, that creates a huge opportunity for theory and for computational methods. Computational methods have a huge potential role to play in filling in the gaps and guiding the experimental understanding of this important class of proteins. And that's one of the things that has drawn us to this field. And in particular, there's a lot of really interesting open questions associated with membrane proteins and how they do, what they do, and how they get where they're supposed to be. So obvious questions, even at this stage in the talk, are, how does this translocon work in governing the passage of matter across the membrane or into the membrane? How does it actually work? You know, the membrane protein itself, you see, has this sort of characteristic looping. It folds and loops across the membrane, assuming a so-called topology. What dictates the onset or the establishment of that particular topology? What governs the establishment of that topology? And given that the expression problem is such an important bottleneck, what can we do to enhance the levels of available protein expression for this important class of proteins? So these are the questions that we want to be thinking about to advance the study of this important class. So fine, we're going to use a theoretical approach. We're going to try to simulate, using a computer, what's going on with these proteins. So how are we going to do it? You might ask yourself, what would be the natural tool that you would employ to try and describe what's going on at the atomic level of this incredibly complicated system? Well, one very natural assumption that you might make is to treat all of the different atoms of the system as a system of classical mechanical objects. We'll just apply Newton's equation to everything, good old force equals mass times acceleration, and we will use these equations of motion, these rules for how the system interacts, and apply that to all the different atoms in the system. So, given that I know where the position is and how it's moving at a particular time, I can calculate all of the different forces on each atom due to all the other atoms in the system. This is not an easy task. It involves approximating Schrödinger's equation, but I can do it. I can calculate those forces. And then I can use F equals ma to figure out the positions and velocities at a later time, delta t. And then I just do this billions and billions and billions of times. And I build up what is called a trajectory, essentially a movie of how the system evolves as a function of time, atom by atom, with all the different atoms interacting with respect to each other. Incidentally, this is also what JPL does, for example, when they shoot a satellite into space, right? But they basically evolve the equations of motion. They look at the trajectory of the satellite as a function of time. Of course, they're doing that for one satellite, and we're doing this regularly for hundreds of thousands of atoms, but there's that. So let's do that. Let's simulate this particular system, and let's look at the classical equations of motion and look at what happens when we make a movie for the time evolution of this protein as it gets inserted in the cell membrane. And this is an actual simulation using over 140,000 atoms. You can watch it. And we're doing, again, billions and billions and billions of those different integrated steps, reaching, for our molecular dynamics perspectives, the Herculean timescale of 4 × 10⁻⁶ seconds. And this required total computation time of 2 weeks on one of the largest supercomputers on Earth. And you can learn a lot from calculations of this kind. You can learn about how things are moving and what's going on, and you can extract a lot of useful information. And again, this was done not just on my laptop, it was done on what's called the Anton supercomputing system, and it was specially designed for exactly calculations of this kind. So this really is the longest and most powerful sort of computing that one can do for trajectories and the simulation of systems of this kind. But you can immediately see that there's a problem. Just one trajectory, one simulation of 10⁻⁶ seconds is on the verge of being computationally overwhelming. The actual amount of time it takes biology to perform this task of synthesizing a protein is more like 100 seconds. So even our best computational efforts using this approach come on the order of 100 million times too short in terms of the accessible timescale in comparison to biology. To put that another way, if I ran this calculation at the current rate to actually reach the biological timescale, it would take almost exactly 1 million years, which is not good news for the graduate students. So what to do? And the obvious thing is to reduce the complexity of the system. What we're going to do is use our insights and use calculations that are carefully designed to simplify the representation of the system. We call this coarse graining because we're reducing the resolution of the system, and to build a model for protein synthesis for this exact process that enables simulations that are on the minute timescales that biology actually occurs. So this is a strategy that I just showed you that does not work using all atom simulations and trying to reach full minutes for the membrane integration process. And here's a strategy that a rather brilliant set of students and postdocs in my group developed over the years to circumvent that challenge. What we've been doing are lots of those relatively short atomic simulations, finding out what the moving parts are, finding out how much energy it takes to move those different moving parts, finding out how that changes when you put different things into the translocon channel, all the different details and factors of the system. And we've used that to build a far more simplified representation for the system that actually makes possible long, minute-long, coarse-grained simulations that enable the efficient simulation of this protein synthesis problem. So what does that look like in practice? So here's the membrane you can barely see. This is the translocon. This is the bottom of the ribosome in our coarse-grained simulation. And we can now actually look at trajectories that correspond to 1 biological minute, but which each can be obtained in more like 10 hours on a single computer processor. And you can see the membrane protein coming out. You can see it establishing its topology, looping around, and eventually you have a 3-spanning protein that comes out. That, again, only took approximately 10 hours on a single processor. And now what we can do is we can actually use this coarse-grained model to directly investigate the process of membrane protein synthesis. And we have the movies. We have the full detail at all times of what's going on in this particular process. So using that, we can return to some of these really interesting questions that we laid out at the beginning. How does the translocon work? What dictates membrane protein topology? And how can we improve expression using what we learn from this particular approach? And what I'm going to first do is focus on this question here: what dictates membrane protein topology? Now, the standard theory, kind of canonical view in the field, and the thing that you might naively assume, is that as protein comes out of the ribosome and becomes a membrane protein, It's got a first bit that comes out, and that first bit chooses a side, and that choice sort of dictates everything ever after. Sort of locks in the overall topology, such that you just get whatever the first little bit chose. Fine, sensible. However, there are proteins that sort of fly in the face of this simple idea. In particular, EMRE is a really interesting membrane protein that suggests a more complex situation. EMRE is what is called a dual topology protein. It means that 50% goes in with one orientation, one overall topology, but the other 50% actually goes in with the exact opposite orientation with respect to the membrane. You kind of get this 50-50 distribution of things that get inserted into the cell membrane. And in fact, this is critically important because these two things with opposite orientation then dimerize with respect to each other. You see the blue and the yellow, which are the same protein just in opposite orientations. They dimerize to form an important channel that allows for the exporting of drug molecules across the cell. So you can see that EMRE is itself an interesting character because it exhibits this sort of 50-50 distribution with respect to the cell membrane. But then there's something really interesting that was also observed about this system. If people make changes to the protein sequence, if you change the actual sequence of the amino acids, even very late in the EMRE sequence, not close to the N-terminus, but right there even at the very caboose of the protein, you can shift this overall balance from 50-50 to things that are far more 90-10, 20-80, and so on and so forth. And this flies in the face of the prevailing view about what is establishing the overall orientation of these systems. If it got locked at the very beginning, then changing something at the very end shouldn't matter at all. So something different is at play. Fine, let's look at that in terms of data. This is actual real experimental data, where what they're able to do is to examine the relative orientation of one topology, which we'll call C in, N in, C in, or N out, C out, and the two halves always add up to 1, so when it's approximately 50-50, that means you have 50% of one of the two orientations. And what they're doing then is they make different mutations on that protein. They change the protein sequence, and they do this in a whole lot of different ways, indicated by those red dots. And what you can see is the bar bounces up and down. It goes much more dominantly in one orientation, much more dominantly in the other orientation, and so on and so forth. And you can see that that happens regardless of whether or not the mutations are close to the beginning of the protein or close to the very end of the protein. So it's very clear that the establishment of this topology of the membrane proteins is much more fluid than one might have initially supposed. Now we can investigate this. Experimentally, that's all we have. The underlying mechanism is totally unclear. We don't know why it's giving rise to this particular behavior. But what we can do is we can then use the coarse-grained simulation approach that I just showed you to reproduce the corresponding experiment. So each one of these bars now corresponds to 1,000 of those protein synthesis trajectories that I just showed you with the corresponding protein sequence. And what we do is when we run those 1,000 trajectories of the system, we ask what fraction arrive at one orientation or the other orientation. And what you can immediately see is that the agreement between experiment and theory for this particular case is strikingly good. In 15 out of the 16 cases, we correctly predict the degree to which shifts one way versus the other way with respect to that 50-50 distribution. And this is really exciting because, as I pointed out earlier, those movies give you the underlying molecular detail. We can then go back and analyze those simulated trajectories to investigate the mechanistic origin of the trends. And we've done that. We've done that in great detail, and I'm not going to go through it in great detail, except to point you to the reference and to make the key points that I really want to make. The ability to perform many long timescale simulations of membrane protein synthesis yields direct comparison between simulations and experiment. It enables us to gain mechanistic insights that would have been inaccessible experimentally. And it enables predictions. We're able to make a number of predictions for new experimental studies based on the underlying mechanistic insights that were obtained. So fine, this tool is powerful. This tool enables us to understand key questions associated with these earliest stages of membrane protein integration. But let's return to this question about membrane proteins being so important, the fact that we want to study, and the fact that there are major bottlenecks associated to that study, associated with the fact that we can't produce enough membrane protein to actually perform the studies that are of interest. Can we use this tool that we've developed to begin to address that problem, as opposed to just investigating mechanistic questions? So to think about that a little bit more, let's turn to how experimentalists actually make membrane proteins. So let's suppose for a second that we're interested in this particular TAT-C protein, shown in terms of its structure and its overall topology, and we're going to worry about the specific TAT-C protein that's made by a particular bacteria, Staphylococcus, and we call that version of TAT-C, that version of the protein that is made by a particular bacteria, the homologue of TAT-C. So this is the Staphylococcus homologue of TAT-C. Each bacteria makes a slightly different version because the bacteria themselves have had evolutionary modifications as a function of time. Fine, so how do we actually obtain that membrane protein experimentally? Well, what one does is one attempts to express the membrane protein in a bacterial host, such as E. coli. What we do is we basically, for a given protein sequence, we take the DNA sequence that encodes that protein, we insert that DNA into the E. coli cells, we culture those E. coli cells, and then we purify the resulting membrane protein. Think about that for a second. It's an incredible thing that this is like a routine thing that goes on in biochemistry labs. They're essentially harvesting proteins from E. coli, essentially the same way that you kind of harvest pearls from oysters. You seed them, you let them grow, and then you scrape out what you want at the end of the day. And that's wonderful when it works, but the whole problem is that sometimes it works, and sometimes it doesn't, and you can't get the particular protein that you want. That's the essence of this expression problem. And to illustrate that challenge, let's look at some actual experimental data. This is data that's obtained in the laboratory of Professor Bill Clemens, fantastic collaborator on this project, and his graduate student, Stephen Marshall. And what we're looking at here is the experimentally observed levels of membrane protein expression of the TAT-C protein, but for many different homologues. So this bacteria makes one version of it, this bacteria makes another version of it, so on and so forth. We try to express all those different proteins, one after the other, in E. coli, and some work, and some don't. Staphylococcus does not, for example. That was our target protein. And you see exactly the problem. It's kind of this scatterboard expectation for what's going to work and what isn't going to work, and there's no rhyme or reason. There's no theoretical way. If I need to study this protein, I have no way to rationally optimize the amount of protein that I can actually get and to study it. Another thing that you see from this data, that even very small changes in the protein sequence. These are, after all, the same protein, just modified slightly in terms of their sequence according to which of the bacteria made them. Even very small changes in protein sequence have a big effect on the observed levels of expression in E. coli. And again, there's no rationale for how to improve that, which is the essence of the problem. So then the Clemens group did something really interesting. If small sequence changes matter, let's try to identify which parts of the protein sequence are having such a big effect on whether or not the E. coli happily make that particular protein. So let's take a protein that works, a winner. Let's take a protein that does not express, feel like a loser. And let's make combinations of this winner and loser, so-called chimeras, and look at the performance of those mixed and matched protein sequences. So this particular one has the first half coming from the NT, homolog, and the second half coming from the AA homolog. These have only one of the different transmembrane domains swapped in. This guy has all the different loops from one swapped into the other. This one, for example, just has the very C-terminus swapped in. And the experimental analysis of many of these different chimeras reveal that extraordinarily small sequence changes, quite surprisingly, have a big impact on expression. For example, you can take this protein right here, which is like 99% MT-TAT-C, which stinks. If you just put the tail from AA-TAT-C on that guy, you recover most of the observed expression. There's something incredibly sensitive going on where small sequence changes have a big impact on whether or not a bacteria can actually make that particular protein. So why? What is the underlying mechanism? Well, they had all this wonderful experimental data, but there was no understanding of what's giving rise to this particular trend. So what we can do is we can, again, use the coarse-grained model that we developed to simulate this TAT-C membrane protein synthesis, and we can investigate whether or not we can see something that is at play. So what we're going to do is we're going to perform, again, thousands of these TAT-C synthesis trajectories. And what we're going to do is we're going to ask what fraction of those simulated trajectories reach the correct overall topology of the membrane protein. And what you see is really quite striking. On the left-hand side is what E. coli makes in the laboratory. It's the amount of these three different protein sequences that actually get synthesized. On the right-hand side is a computer simulation of what fraction of time when the protein comes out of the membrane does it loop through the membrane, assume the correct topology. What fraction of time does it assume that correct topology? And you can see that the trend is actually quite striking in terms of its agreement. And the basis for this, although this was an incredibly surprising result, is itself really quite sensible. If the membrane protein adopts the correct topology, as predicted over here, this has the opportunity to be a correctly folded and stable protein. If the protein does not assume the correct topology, as predicted for this MT-TAT-C case, because the C tail is incorrectly integrating into the cell membrane and basically flopping her out incorrectly, this wrong topology proteins are identified by the cell, and therefore you don't actually see them produced in the E. coli. These ones are basically being degraded by the cellular machinery, leading to that dramatic reduction in the overall expression level. So this actually creates remarkable conclusion and actually a really exciting opportunity as well. The first is that this, as well as, of course, a huge number of an additional set of simulations and experimental studies on a range of different sequences, enabled us to establish that the integration efficiency, the fraction of time that proteins loop into the membrane with the correct overall topology, is a major bottleneck for membrane protein expression. Now, return to the picture of the subway system. Imagine all the different things in that congested environment that could be going wrong. Imagine all the different steps of making a protein that could influence in a cellular environment whether or not that protein is going to be survived, whether or not it's going to be made, whether or not it's going to be seen and expressed. The fact that it has to do with this particular step of looping into the membrane was a very surprising and very exciting identification of the source of the problem. And it comes with it an important corollary. If the correct integration is the key bottleneck to expression, and if we can computationally predict the amount of correct integration, it stands to reason that we should be able to predict changes in the protein sequence that will improve the level of integration and therefore improve the level of expression. We have an opportunity to actually design new proteins that the cell will be happy to make in greater abundance than would have been the case by just choosing things randomly out of a hat. So that's what we set out to do. This is Mikhail Neeson, this is Stephen Marshall, graduate students in my lab and in Bill Clemens' lab. What we did is we made over 100 predictions of different homologs of TAT-C with respect to whether or not they should increase the level expression or decrease the level expression based on these computational predictions. And then Stephen went into the lab and actually measured all of those different predictions. And with this large number of things, we can actually do meaningful statistics, gathering false positives, true positives, true negatives, false positives, and false negatives. And one can do a statistical analysis to obtain the likelihood that we correctly predict whether or not a particular change in the protein sequence will lead to enhanced expression or not. And what we're able to demonstrate is that these coarse-grained simulations are over 3 times more likely to be able to return sequences that lead to improved membrane expression than would be the case if, as was the previous modus operandi, we just had to make random mutations. It provides actually a computational tool that we can use to guide protein mutations that will lead to enhanced levels of expression. You might ask, how does this particular predictor of membrane protein expression compare to other things that you might use to predict that level of expression? So for example, a membrane protein has many different attributes. It has loose links, it has hydrophobicity, it has charges. Many different things have been proposed in the literature as being good predictors for membrane protein expression. And what we're actually plotting here is the performance, according to 2 different measures of statistical correlation, the performance of those alternative prediction tools versus our simulated integration efficiency tool, the coarse-grained simulations tool, is in serving as a predictor for experimental levels of expression. And the point here is that the simulations, these coarse-grained simulations that we've been able to develop, are a better predictor than other previously proposed features of the membrane protein sequence by a significant measure. So it's really exciting, actually. What we've been able to do is to build simplified computational tools that enable us to actually look at the initial key steps of membrane protein synthesis. We've been able to understand what dictates the establishment of membrane protein topology, the way in which it loops into the cell. And we've been able to use that to identify a connection between the establishment of that topology and the observed levels of membrane protein expression. And that's truly exciting because this provides a computational avenue to removing a major bottleneck. If we can turn this 5 to 10% to 15 to 30%, that opens up a large number of new protein systems that are major drug targets that we'll be able to then have enough to actually perform biochemical studies, drug targeting studies, structural studies. And this is exactly what our group is in the process of doing moving forward. So what I hope I've been able to show you is that there's 3 different things. The first is that at the molecular level, we've seen that the cell is kind of a wild and woolly and extraordinarily interesting place. It's crowded and congested, but the fact that it preserves the fidelity and the organization that it does is a truly amazing thing. What I hope that I've been able to show you is that computational approaches, things that are based on the fundamental equations of motion, Newton's equations, Schrodinger's equations, can be brought to bear in a way that is truly new in the understanding of these biological processes, providing improved understanding, improved prediction, and indeed, as you saw, the design of improved cellular behavior based purely on computational prediction. And we're very excited about the possibility of removing bottlenecks in the expression of membrane proteins. And by doing so, we hope to advance the study of these important cellular gatekeepers that control the flow of matter and material across the cell membrane. So with that, I'd like to thank the people who did the work. I've been lucky to have an extraordinary array of really fantastic graduate students and postdocs in my research group. Bin Zhang was a graduate student who pioneered the early stage of the development of this coarse-grained model. Just 2 weeks ago, he accepted a faculty position at MIT in the chemistry department, so he's going to the other coast and is now the competition, so he'll be banished from our good favors. Mikhail Nielsen is a graduate student who's been involved in all aspects of this work, including particularly that membrane protein expression work that I showed you a moment ago. Reed Van Lane is a postdoc, a former postdoc in my group, who did the topology studies with EMRE. He also recently left the group for a faculty position in the chemistry department of the University of Wisconsin. And Connie Wang is a fantastic graduate student that's been involved in all aspects of the model development. As you saw, a lot of this work hinges on the ability to collaborate with experimentalists and to, in a rapid manner, predict and test those predictions experimentally in the lab. And we've enjoyed weekly group meetings with the Clemens group where we actually can iterate this theoretical experimental cycle on a rapid timescale. And that's been an extremely rewarding and fun aspect of this overall project. And Stephen Marshall's been the graduate student who's been leading those efforts in practice. I'd like to thank the members of my group present and past. Again, it's been a truly fantastic group of people. Many have gone off to great faculty jobs in other excellent institutions. I'd like to thank the range of different Caltech collaborators that we don't necessarily involve this particular project, but that are involved a number of other different projects that are going on in the group. I'd like to thank the funding that makes this possible, as well as the supercomputing resources upon which we actually rest in terms of performance of this sort of work. And with that, I'd like to thank you for your attention and be happy to answer any questions.